All right, so this section is going to quickly go over the PowerPoints for the cardiovascular system. And the first thing to remember is always the functions of the system. So, and the organs that belong in the system. So in the case of the cardiovascular system, the organs are going to be the artery, the blood vessels themselves, which will be arteries and veins. It is important to remember that arteries are the blood vessels that take blood away from the heart. Away, if that helps you remember. On the other hand, the veins are the blood vessels that bring blood to the heart. Capillaries are what we call exchange vessels. These will be talked about in chapter 21 a little bit more in detail. So we're not going to worry about those too much right now. The other organ that belongs in the cardiovascular system is the pump of the cardiovascular system, which is the heart. And notice that it says it is a two-pump system. And then obviously blood. Okay, so one of the characteristics of the human heart is that it functions on a two-circuit system. The function of the heart is going to be to pump blood that is arriving, the oxygenated blood that is arriving at the right side of the heart, and it is going to, that deoxygenated blood will be pumped to the lungs to be oxygenated. This is called the pulmonary circuit. Pulmonary means lungs. Blood is oxygenated in the lungs, and it will come back on the left side of the heart once it's oxygenated, and from there it will be pumped to the rest of the body. This is the systemic circuit. So you should be able to distinguish between the pulmonary circuit and the systemic circuit. Again, the pulmonary circuit consists of the blood vessels that bring blood to the heart. Blood arrives at the right atrium, which is the top right chamber of the heart. From there, blood, this is color in blue because it represents blood without oxygen, the oxygenated blood. The oxygenated blood is then transferred to the right ventricle, which are the lower chambers, and from there it's going to be pumped out through the pulmonary trunk, and which divides into the right and left pulmonary arteries, taking blood towards the lungs on both sides. In the lungs, blood will be oxygenated, so oxygen will go into the blood vessels. CO2 will be delivered out into the lungs to be breathed out. This oxygenated blood now returns to the heart on the left side. So it's going to arrive. Now this is blood arriving, so the blood vessels will be veins. It's going to arrive via the pulmonary veins to the left atrium of the heart, from where blood will be transferred to the left ventricle, and from there will be pumped out of the left ventricle through the aorta. It will be delivered to the rest of the body. Notice that this blood is painted red to represent oxygenated blood. This is the systemic circuit. So the pulmonary circuit takes place on the right side of the heart, and the deoxygenated blood is pumped to the lungs, where it is oxygenated. The systemic circuit takes place on the left side of the heart when the oxygenated blood returns, and from there is pumped out through the aorta to the rest of the body. So definitely know the components of the pulmonary circuit and the circulatory and the systemic circuit. This is another depiction of the same concept. I'm going to skip through the questions, but essentially it's going to tell you the same things that we're talking about right now. Now, as far as the anatomy of the heart will be tested in the practical exam, but you should know the anatomy also for lecture, because the functions of the anatomical structures will be the theme of the lecture exams. So just as a introduction, the heart is a triangular shaped organ. It has a base, and the base of the heart is on top, and it is the place where blood vessels come in or out of the heart. The taper end is called the apex, as at the bottom, and notice that it is tilted towards the left side of the body. The heart is located between the second intercostal space and the fifth intercostal space. Again, it is in the middle of the thoracic cavity. However, it is tilted towards the left. The space where it's located is called the mediastinum. And let's see, what else? Yeah, this is showing you the location of the heart. Notice that it's right on top of the diaphragm and is wedged in between the two lungs, which makes sense because the blood has to be pumped to the lungs to be oxygenated. So it makes sense for the lungs to be very close to the heart. The heart is enveloped in a serous membrane. This serous membrane is called the pericardium. The pericardium has these collagen fibers that are going to attach the lung to adjacent blood vessels and to the diaphragm below. The serous pericardium is the membrane itself. And if you remember, serous membranes were made of two layers. It was a two-layer membrane. The parietal pericardium is the outer layer adjacent to the fibrous pericardium. The parietal pericardium is attached to the external structures around the heart. On the other hand, the visceral pericardium is the serous membrane that touches the heart. It is also called the epicardium. So again, you should just know the names of these two membranes. The epicardium or visceral pericardium touches the heart itself, envelops the heart itself. The parietal pericardium is the outer layer of the membrane. In between, there is a cavity, which is called the pericardial cavity, filled with pericardial fluid. The fluid cushions and reduces friction as the heart pumps. Okay. The heart consists of three tissue layers. The outermost layer is the serous membrane or visceral pericardium, also called the epicardium. Remember, epi meant upon, so upon cardium, upon the heart. The wall of the heart is made of muscle, cardiac muscle. It is called the myocardium, and this is the thickest part of the heart. So this is cardiac muscle. And then lining the heart chambers, muscle, okay. Lining the heart chambers is the endocardium, which is a thin membrane of simple squamous epithelial tissue. Very much the same as the mesothelium, the mesothelium, which lines the blood vessel. So this is simple squamous epithelial tissue. Okay. All right, so you should definitely know these three membranes, the epicardium, the myocardium, which is the thickest layer, and then the endocardium, which is simple squamous epithelial tissue. This is a depiction of the epicardium right there. You can see the membrane as it's being pulled away. Okay. A condition in which there's an inflammation of the pericardium is called pericarditis, and inflammation results in fluid buildup. The fluid builds in the pericardial cavity, accumulating and putting pressure on the heart and causing what's called cardiac tamponade. So cardiac tamponade is the large volume of fluid or blood accumulated in the pericardial cavity. When we look at the surface anatomy of the heart, meaning the external anatomy of the heart, remember the top chambers are called the atria, the bottom chambers are called the ventricles. The atria on the surface has extensions of the chamber, which are called the auricles. So these are anterior extensions of the atria. Between the atria and the ventricle, there is a groove that goes around. That groove is called the coronary sulcus, which represents the separation between atria and ventricles. So the separation between the top chambers and the bottom chambers. In between the right and left ventricles, there is a wall which can be distinguished on the surface by a sulcus. This is the interventricular sulcus. This is the anterior and posterior sulcus that separates the right and left ventricles. So here we can see the anterior interventricular sulcus, we turn the heart around and we'll see the posterior interventricular sulcus. Again, this is a wall that represents the outside part of a wall that separates both ventricles. Okay, so that's more of the same. Yeah, notice that this sulci, which is a plural for sulcus, is covered by fat tissue and blood vessels. So you can see blood vessels and fat tissue in these areas. Again, another depiction of the heart, and you can see the anterior interventricular sulcus right there, and then the coronary sulcus right there. One of the important points that you need to remember when you look at pictures of the heart is to pay attention whether you're looking at an anterior aspect of the heart or a posterior aspect of the heart. In this case, it is anterior. You can clearly see the right atrium. The left atrium is a little bit more difficult to see. You definitely see the aorta, the aortic arch, and you can see the pulmonary trunk. A posterior view of the heart, right here, allows you to see the left atrium a lot better. You can see, now you do not see the pulmonary trunk, but you see the two blood vessels that come from the pulmonary trunk, the left and right pulmonary arteries. Okay. You can see also the pulmonary veins as they arrive at the left ventricle, which is something you did not see very well from the anterior view. So this is a posterior view. Make sure when you look at a heart, ask yourself whether you're looking at it from a posterior or anterior view, because that will vary the types of structures that you see. Okay. Now, moving on, we're going to talk about the 4 chambers of the heart. Let's see. We have 2 chambers on each side. The chambers are completely separated. There is no connection between the right side and the left side. The superior chambers are called the atria. So we have a right atria and a left atria. The function of the atria is to collect blood from the veins that arrive from other parts of the body and deliver blood into the atria. So the job of the atria is to collect venous blood and then to pump that blood or to deliver that blood to the lower chambers, which are the ventricles. The ventricles are inferior. They're very thick, muscular chambers. Their job will be to pump the blood through the arteries that lead to the lungs in the case of the right ventricle and to the rest of the body in the case of the left ventricle. Remember that the right and the left side of the heart are independent from each other. They're completely separated. In the top layers, the top chambers are separated by the interatrial septum. The bottom chambers are separated by the interventricular septum. This model is showing you the interatrial septum right here. The bottom septum is the interventricular septum. You can see the right atria and the right ventricle completely separated from the left atria and the left ventricles. Okay, we're going to look at each of the chambers independently and note the types of structures that we see in each. So first, we're going to look at the right side. So now we're looking at the right atria. And again, the function of both atria are to collect venous blood. In the case of the right atria, there are 3 blood vessels that arrive at the right atria. So we have a little heart here. So we're looking at the right side. And notice the right side is always painted blue because it's receiving deoxygenated blood. So there are going to be 3 blood vessels that arrive at the right atrium. We're going to have the superior vena cava, the inferior vena cava, and the coronary sinus. All three vessels are bringing the oxygenated blood to the right atrium. Okay. Another feature of the right atrium is a remnant of a fetal heart. In the fetus, the right atria and the left atrium are connected through a hole which in the fetal heart is called the fossa ovalis. So the fossa ovalis, or the foramen ovalis, foramen ovalis. The foramen, remember foramen from last semester, main hole. The foramen ovalis is this hole that is connecting the right atria and the left atria in the fetal heart. And we'll talk about the function of it here in just a second. Once the person is born, the foramen ovalis closes, and what remains is a little indentation called the fossa ovalis. Okay. Another feature of the right atria is these muscle ridges on the anterior aspect of the wall of the atria called pectinated muscle. I'll show you a picture in just a second. Two other features unique to the right atria. This is only found on the right atrium, I should say, atrium which is singular, in the right atrium are going to be the SA node and the AV nodes. The SA node is called the pacemaker of the heart, and it is the area of of nodal cells, little specialized cells that initiate the action potential. So that is located on the superior right corner of the right atrium. From there, the AP, the action potentials rated, are sent to the AV node located on the inferior left corner of the right atrium. That's the AV node. It is worthy to note these structures are microscopic. Therefore, they're not going to be depicted in models. So this is a model of the heart. This is the right atrium. So let's go over these structures we talked about. So we have the superior vena cava, inferior vena cava, and coronary sinus, bringing blood into the right atrium. The right atrium is going to have the fossa ovalis, which is what remains of the fetal foramen ovalis. The SA nodes and AV nodes are microscopic. We cannot see them in these kinds of depictions, but if we could see them, they would be on the top right corner of the right atrium. That would be the SA node. And on the bottom left corner of the right atrium, we'll have the AV node. Okay. All right, so this is just repeating the types of blood vessels that are on the right side. This is showing you what we call a patent foramen ovale. Okay, so the foramen ovale is the actual hole that is seen in the fetal heart. This is an opening that connects the right and left atria in the embryo and the fetus. This foramen is supposed to close upon birth, leaving behind a fossa ovalis. It's a little depression that represents what used to be there. If it does not close, that would be an abnormal condition. And what happens to these babies is that the deoxygenated blood from the right side travels to the left side and it mixes with the deoxygenated blood. So now the blood that is being pumped out to the body is going to have less oxygen. So the babies are lacking oxygenation. When you think about it, let's do this again. In a fetus has lungs that are not yet working. Therefore, there is no need for the right side of the heart to pump the blood to the lungs. There is no need to go to the lungs. The lungs are not working. The lungs are not oxygenating blood. Therefore, instead, in the fetal heart, the oxygenated blood is transferred to the left side, and from there it will be taken to the placenta, which is the place that is going to oxygenate the blood. And so that blood has to be diverted away from the lungs towards the left side to be pumped out into the aorta towards the placenta to be oxygenated. So that is the purpose of the foramen ovale in a fetus. Once the person is born, now obviously the placenta is not, it's removed, and now the lungs have to take over and begin oxygenating. So the foramen ovale has to be closed to now divert blood to the lungs. Okay. Remember, we talked about another feature of the atria, right and left atrium actually, is this pectinated muscle, which are ridges of muscles on the anterior wall of the atria. Here's this pectinated muscle right there in the atria. So these are ridges of muscles. And a depiction of the pectinated muscle right here. Okay. All right, now let's move on to the left atrium. The left atrium receives openings from 4 veins, 2 on the right side, 2 on the left side. They are all 4 of them called pulmonary veins, and they bring oxygen-rich blood directly from the lungs. So this is the blood that has just been oxygenated. The left atrium also has pectinated muscle. Notice that it does not have nodes. So the nodes are exclusive to the right atrium. So here's a depiction of the right and the left pulmonary veins bringing blood to the left atrium. These right and left pulmonary veins are better seen from the posterior view of the heart. You can really see all 4 of them clearly. All right, and now we're going to move on to the ventricles. The ventricles receive the blood from their respective atria, and they're going to pump the blood out through arteries. On the right side, when the right atria contracts, it's going to pump blood to the pulmonary trunk. Another feature of the right atria are these huge ridges of muscle. The equivalent of the pectinated muscle in the atria is now called trabeculae carneae in the ventricle. The ventricles also have these finger-like muscle projections called papillary muscle. So when you look at the right ventricle, the right ventricle is receiving the oxygenated blood traveling from the right atrium. The ventricle contracts and is going to pump that blood out through the pulmonary trunk to be delivered through the pulmonary arteries to the right and left trunk. On the left side, we're going to see much of the same structures. The left side also has the trabeculae carneae, which are the muscle ridges, the equivalent of the pectinated muscle in the atria. The left ventricle also has these finger-like projections of muscles called the papillary muscles. The left ventricle pumps blood, oxygenated blood, out through the aorta, which is the largest vessel in the body, and the aorta is going to take this oxygenated blood to the rest of the body. This is the beginning of systemic circulation. The aorta arches, forming the aortic arch, and then it continues on the back of the heart, making the descending aorta. This is a depiction of the trabeculae carneae, the ridges, muscle ridges, which are found in both the right and the left ventricles, and a depiction of the papillary muscles right here. Papillary muscles are these finger-like projections of muscles, which, let me show you their function here in the next picture. Papillary muscles are right here. You can really see them in this picture of the left side of the heart. You can see these finger-like projections are the papillary muscles. Notice that the papillary muscles are attached to these cord-like structures right there. They are called the chordae tendinae. the chordae tendineae in turn are attached to a valve that can open and close between atrium and ventricles. So the papillary muscles are attached to the chordae tendineae. Chordae tendineae are attached to the atrioventricular valves. This same thing is happening on the right side. This is the papillary muscles, this is the chordae tendineae, and this is the AV valve on the right side. There is obviously a function that explains this construction, which we're going to talk about here in a little bit. Okay, this is a posterior view of the heart, and you can see the left ventricle right there. Notice we don't really see the aorta from this view. We see it as it arches up here. Let's talk about these valves between atria and ventricles. The valves are called atrioventricular valves. On the right side, the atrioventricular valve has three parts, three cusps, so it's called bicuspid valve. On the left side, the atrioventricular valve has two cusps, so it's called the bicuspid valve. Its other name is mitral valve. So you really have to know both names on the right side. Bicuspid, I'm sorry, on the left side. This is the left side right here. Okay. So on the left side, we have the bicuspid or mitral valve. On the right side is the tricuspid, or that actually just has one name, tricuspid. Now, the entrance into the pulmonary trunk and the aorta is guarded by valves. These valves are called semilunar valves. On the right side, the pulmonary semilunar valve guards the entrance into the pulmonary trunk. On the left side, the aortic semilunar valve guards the entrance into the aorta. You should be able to recognize these valves from a picture or a model of the heart. So here you have on the left side, the mitral or bicuspid valve. On the right side, the tricuspid valve. Notice the chordae tendineae attaching to these valves and in turn attaching to the papillary muscles. It says that the papillary muscles prevent opening of the valves into atria during ventricular contraction when ventricles are filled with blood. We're going to see this here in just a second. Okay. So again, I'm skipping through these questions. The next section is going to look at again, blood vessels of the heart. You really have to know these. You have to know that on the right side, the superior and inferior vena cava and the coronary sinus are arriving, and that blood is sent to the right ventricle and then goes out through the pulmonary trunks. On the left side, the pulmonary veins arrive on the left atria. From there, the blood is transferred to the left ventricle and it leaves through the aorta. So you should be able to recognize these blood vessels coming in and out of the heart. Now, even though the heart is pumping blood through the chambers and blood is circulating or is found in these chambers, that blood is not feeding the heart tissue. It's not feeding the myocardium. So the heart tissue needs its own blood vessels to feed it. Okay. This is what we call the coronary blood vessels. On these coronary blood vessels begin on the right and the left by the right and left coronary arteries. So these are tiny arteries that branch from the aorta. These are the first vessels to branch from the base of the aorta. They're going to supply blood to heart tissue, especially the myocardium, and they're going to travel through coronary sulcus. So let me show you where these blood vessels are located. So this is an anterior view of the heart. Here is the aorta as it exits the left ventricle. And notice that the very first little branches that come out at the base of the aorta are going to be the left coronary artery and the right coronary arteries. Later on, larger vessels are going to branch from the aorta. On the left side, the left coronary artery is actually very short, almost immediately branches into two other blood vessels. One of them is going to travel down the interventricular septum, and so this blood vessel is going to be called the anterior interventricular artery, which is going to be feeding the left ventricle. The other branch of the left coronary artery is called the circumflex. We don't have it labeled here, but it would be that one there, circumflex. And it's called circumflex because it travels through the coronary sulcus, and it kind of encircles the heart on the left side, feeding the posterior aspect of the atria and ventricles. So again, on the left side, the left coronary artery is going to split into the anterior interventricular artery and the circumflex artery. We're just going to ignore the left marginal artery because it's not part of the list of vessels you have to know. On the right side, the right coronary artery is much more pronounced. It's longer. It goes out and begins to travel through the coronary sulcus to the backside of the heart, and then branches down into a artery that travels through the posterior interventricular septus. So this artery is called the the posterior interventricular artery, which we don't have it labeled here. No. Okay, so this will be the posterior interventricular artery. We'll see it in the next PowerPoint, but let me go ahead and write it down in here. Posterior interventricular artery as a branch of the right coronary artery. Okay. All right, another branch that you need to know of the right coronary artery is this little branch right here that is on the side of the heart. It's called the right marginal artery, feeding the lateral aspect of the heart in that area. So the two branches that you need to know of the right coronary artery are going to be the right marginal artery and the posterior interventricular artery. So there's the posterior interventricular artery. It is labeled in this picture. All right, when it comes to veins, these are the blood vessels that collect deoxygenated blood, poor blood from the tissues of the heart, again, mainly the cardiac muscle. So now this blood has to be returned to the heart for oxygenation. And remember, deoxygenated blood arrives at the right atrium. So this blood is going to arrive at the right atrium. The large blood vessels collecting these deoxygenated blood from the heart are going to be the great cardiac vein, which is collecting blood from the left side of the heart. I'll show you where it is located in just a second. This small cardiac vein that drains from the margins of the heart on the right side, and the middle cardiac vein that drains blood from the posterior aspect of the heart. All these blood vessels eventually are going to deliver their blood to the coronary sinus. And remember, this is the sinus, this is the vein that arrives at the right atrium. So let's look at the location of these blood vessels. This is the great cardiac vein right here, which shares the same space, the same area, the anterior interventricular sulcus, the same location as the anterior interventricular artery. The small cardiac vein right here is in a similar location as the marginal artery. The middle cardiac vein back here is on the back of the heart, sharing the same location as the posterior interventricular artery. Notice these blood vessels take blood to the coronary sinus right here, which dumps its blood into the right atrium. Many of these blood vessels join together, creating circuits of irrigation. that are going to assure that all areas of the heart are always irrigated with blood, even if one area is blocked, other areas continue to irrigate. These connections of blood vessels are called anastomosis. So this is typical of the heart. When it comes to the contraction of the heart, one thing to remember is that the atria contract first in unison. So both right and left atrium contract together. So both atria contract at the same time. Then there is a short period of rest, and then immediately following, both ventricles are going to contract at the same time, right and left. Then there is a long period of rest for both atria and ventricle, and then the whole process begins again. Atria contract, then the ventricles contract, then both chambers rest, then again atria contract, then ventricles contract. The coronary blood vessels deliver oxygen to the heart muscle. The amount of oxygen delivered to the heart muscle is very high compared to the amount of oxygen delivered to all other kinds of muscles in the body. 70% of the oxygen circulating in the arterial coronary blood vessels will be delivered to the muscle of the heart. Compare that to 25% of oxygen circulating through the systemic arteries that is delivered to skeletal muscle. So that tells you that cardiac muscle needs a lot more oxygen because it requires energy to constantly pump. Now, if you have read the objectives for this module, you would have discovered that one of the bonus questions for lecture test number 2 is going to ask you to explain the circulation of blood through the heart from right chamber to left chamber, noting every single blood vessel, chamber, and valve that you encounter. So let's go through it one time, and then I'll show you a PowerPoint where, again, you can see this information. So on the right side, blood is going to arrive via the superior and inferior vena cavas and the coronary sinus. Do not forget coronary sinus. Everybody forgets coronary sinus. From there, blood is going to travel through the tricuspid valve to arrive at the right ventricle. When the right ventricle constricts, blood travels through the pulmonary semilunar valve into the pulmonary trunk. The pulmonary trunk splits into left and right pulmonary arteries, which go to the left and right lungs for oxygenation. Once blood is oxygenated, it comes back from the lungs via the left and the right pulmonary veins, which arrive at the left ventricle. The blood is then pumped through the bicuspid valve into the left ventricle, from where it's going to be traveling through the aortic semilunar valve into the aorta to be pumped and delivered to the rest of the body. This is showing you what happens as blood travels from the atrium into the ventricles. This is a depiction of what happens on the left side of the heart. Notice that when blood is being pumped from the atria into the ventricles, the AV valves, the atrioventricular valves, are open to allow the flow of blood from one chamber to the chamber. On the other hand, the semilunar valves are going to be closed. There will be no blood being pumped out of the ventricles when the ventricles are being filled with blood. So the semilunar valves are closed. The atrioventricular valves are open when blood is traveling from atria to ventricles. On the other hand, when the ventricles pump blood out, when the ventricles begin to constrict, the pressure exerted by the contraction of the ventricle is going to push the blood against the AV valves and is going to close them. At the same time, the same pressure is going to push against the semilunar valves and open them. As the AV valves are being closed, the papillary muscles constrict, and as they constrict, they pull on the chorda tendineae, locking the AV valves. So that at this point, it doesn't matter how much pressure is exerted on the blood as the ventricles constrict, pressure should not open the AV valves, which will remain locked while the ventricles constrict. The semilunar valves have no chordae tendineae, so the pressure is going to open them, and blood is going to be rushed out into the aorta or the pulmonary trunk, depending on which side of the ventricle you're looking at. So this is showing you that during ventricular contraction, as blood is being pumped out through the arteries, aorta or pulmonary trunk, the semilunar valves will be open while the AV valve will be closed. This is the PowerPoint that again shows you the circulate through the heart. Down here you can follow through and note every chamber and every blood vessel and every valve that blood flows through the pulmonary circuit and the systemic circuits. So you should know these, hopefully, if you want to correctly answer that question in the test. Okay. Moving on to other matters, this here is a depiction of what we call heart skeleton. The heart skeleton is the layers of valves, and valves are made of connective tissue. So these are the layers of connective tissue that offer an electrical insulation between the atria above and the ventricles below, so that when the action potentials travel through the atria, the action potentials are not going to begin traveling immediately into the ventricles, but rather, since the atria and the ventricles are electrically insulated by these connective tissue, the action potentials are going to have to be to travel from one part of the heart to the other in a very controlled fashion. This is important because it's allowing the atria to contract first, then there will be a small delay, a short delay, before the ventricle contract next. So these connective tissue skeleton between atria and ventricle is important in insulating both sets of chambers and preventing the whole heart from constricting at the same time, which it would make it disastrous because then it wouldn't work properly. In order for the heart to work properly, the atria contract first, then the ventricles need to contract next as the atria relax. So this again is depicting the skeleton of the heart, which creates an insulation between between atria and ventricle. Okay. The next picture is depicting cardiac muscle. If you remember, cardiac muscle was made of branching muscle fibers with one single nuclei in the middle. This was a striated muscle, meaning that there will be sarcomeres creating traction. You can see the sarcomere right there. Also remember that cardiac muscle was unique because it had intercalated discs, which were these darker structures right there in between cardiac muscle. And the intercalated discs are actually gaps or connections between cardiac muscle cells, which allow from sodium ions to travel from one cell to the next, sending electrical signals along one cell to the next. Okay, let's see. Cardiac muscle cells come in two different populations. We have one set of cells called contractile cells. These are what we call the most numerous cells. These are the ones that are going to constrict and cause the contraction of the chambers, whether it be the atria or the ventricles. The population of cells are not as numerous, but they're very, very important. These are called conducting cells. These are very special cells. These are the cells that initiate action potentials, which are going to be transferred to the contractile cells, stimulating contractile contractions. Now, this is the deal. Think about these. The skeletal muscles that were seen last semester were these muscles constrict when the nervous system sends a signal to the muscle and creates an action potential in the muscle, which will cause action potentials to travel through the muscle cell, cause the release of calcium ions. Calcium ions will initiate contraction in the sarcomeres. This is what happened in a skeletal muscle. This muscle is dependent on the nervous system for its contraction. In contrast, cardiac muscle does not depend on the nervous system for contraction. Instead, conducting cells, or also called nodal cells, are formed very, very early in embryonic development, about 2 weeks into embryonic development. These nodal cells that are going to be part of the heart are formed. And nodal cells, the minute they're formed, they begin to generate their own action potentials. And in doing so, they create sodium currents. Sodium currents are going to be transferred from one cell to the next, depolarizing the cells as these ions travel from cell to cell. This is what's going to create the signal that will contract the contractile cells, the muscle of the heart. Okay. Now, these conducting cells are located at specific places in the heart that we called nodes. So we already talked about the SA node, the upper right side of the right atrium, and the AV node on the lower left side of the right atrium. The SA node constitutes the pacemaker of the heart. These are the group of cells that set the heart's rhythm of contraction and are constantly initiating action potentials or contraction signals. The nodes are the passageway for these action potentials from atria to ventricles. So this is the location in which the action potentials will be transferred from the atria to the ventricles. Notice that the action potentials start out at the right atrium, and from there it will have to be delivered to the rest of the heart. This is what we call the conduction system of the heart. So let's go through conduction system of the heart. Again, it all starts at the right atrium, at the SA node location, which these little cells are constantly creating action potentials at a rhythmical pace, between 70 to 80 action potentials a minute. That is the beats per minute of or the heart rate. And we'll talk more about that in just a second. So the SA nodes create these action potentials or currents of substance, which will be transferred via nodal cells into another node called AV node here. Okay. As these action potentials are traveled from one set of nodal cells to another set of nodal cells, they are also begin to travel out of these nodal pathways into contractile muscle cells, making these cells initiate their contraction. These AP action potentials also cross over to the left area and begin to be transferred to the contractile cells of the left atria. These little cells are very important, and they are somewhat different from the contractile cardiac muscle cells. First of all, nodal cells, and these are also written in another PowerPoint earlier. Nodal cells are larger than contractile cells. So these action potentials travel very quickly through these cells. Okay. All right, so these are larger cells than contractile cells. And they're in charge of delivering these action potentials, the other cells that are going to actually contract, the contractile cells. Now, think about these. The atria need to contract first. Following atrial contraction, there's a very short rest period while the signal is being transferred to the ventricle for ventricular contraction after atrial contraction. So there is a delay between the passage of the action potential between atria and ventricles. It is important to know that that delay is caused by the AV node. So the AV node is going to delay the signal from atria to ventricles. That delay allowed the atria to contract first, and then later the ventricles. So let's talk about that one just a little bit more. Make another point here. I'm going to show you this next picture. Again, the next picture is showing you the conductive system of the heart, which starts at the SA node. The conduction transfers the AV node via these nodal pathways. Also notice that the arrows are showing you that these pathways are taking the contraction to the left atria, and the atria will come first. While the atria is constricting, the signal is traveling to the AV node. The AV node has two features that are going to delay the signal. One feature is that the cells of the AV node are actually small cells. We said nodal cells were larger. However, in the case of the AV node, there's an exception. These cells are actually smaller, and that causes a delay in the signal because small cells take longer for the signal to travel. I don't know what's going on here. Let me just go ahead and erase this again. Okay, come on. This is not working. Give me just a second. Okay, now. All right, let's try this one more time. I need to write down the two features of the AV node that create the delay. So AV node has smaller cells. Small cells create friction, and it slows down the signal. The other feature is that the AV node cells have few intercalated discs, so there are fewer between cells. So again, these current of sodium ions are going to have a hard time traveling from cell to cell. So that delay the signal, giving time for the atria to constrict first. Once the signal leaves the atria, it travels through the AV bundle. Let's see, AV bundle, also bundle of His. So it has two names: AV bundle or bundle of His. The signal will then, the AV bundle is splits into bundle branches, and bundle branches then deliver the signal to Purkinje cells, which are found in the papillary muscles. So that the first cells to constrict in the ventricle will be the papillary muscle cells. Okay, so let's do this again. First nodal cells that create the signal, the pacemaker cells, the pacemaker of the heart, are the cells of the SA node. From there, the signal will travel to the to the AV node, and from there, the AV node is going to delay the signal. And there are two reasons why the AV node delays the signal. One would be because the cells are small at the AV node, and they have few intercalated discs. So that's going to delay the signal. Eventually, it will exit the AV node and travel to the AV bundle. From there, it will travel through the bundle branches. And from there, the action potential will travel to the Purkinje cells in the papillary muscles. Papillary muscle contraction will precede contraction of the rest of the ventricle, meaning very first part of the ventricles that contract will be papillary muscles. That makes sense if you think about it, because when the papillary muscle contracts, it's going to pull on the chordae tendineae, and now it's going to lock the valves shut close, to prevent the blood from returning to the atrium. Now, when the ventricles constrict, blood has nowhere to go except outside. Go out through the spinal valves, through the aorta, and pulmonary trunk. So it is important that these cells in the papillary muscle be the first ones to contract within the ventricle. One more time, the reason why the signal is delayed at the AV node, AV node, is due to the smaller cells at the AV node. That's a mistake right there. Hopefully I have corrected it in the new PowerPoint. And because the AV node has fewer intercalated discs. I'm going to check on these, but I do think I corrected this mistake on the PowerPoints that are posted on eCampus. So you should know the reasons for the delay. Okay, so the delay at the AV, I keep saying bundle, is actually node. Allow atria to constrict before the ventricle. Papillary muscle contraction is going to precede ventricular contraction, locking the AV valves and preventing blood from going back to the atrium. Okay. So now at this point, what we need to look at are the way in which action potentials are created on both the contractile cells and the nodal cells. This right here is the AP, action potential curve, that we saw last semester for skeletal muscle. And this contraction, the AP curve, consisted of depolarization due to the influx of sodium ions, reached a peak at plus 30 millivolts. At that point, sodium voltage channels closed, potassium voltage channels open, and now there was a loss of positive charge via potassium loss, which repolarized the cell, made it back negative. There was a hyperpolarization point, and then the cells went back to rest. Contrast these with the curve that we see in contractile heart cells. Here you have depolarization to the opening of sodium channels. Nothing different here. Depolarization reaches the max of 30 millivolts, just like in a skeletal muscle. From there, the cell begins to repolarize as the potassium channels begin to open and the sodium channels close. So now we have the early repolarization right here, because again, potassium channels open, sodium channels have closed. So now there is a loss of positive charges, making the cell more negative. However, notice that all of a sudden, that repolarization stops and the cell reaches a plateau. The reason for this plateau is because calcium channels all of a sudden have opened. These channels are slow to open, and now you have an influx of calcium into the cell. Remember, we need calcium for muscle contraction, but in this case, the calcium that is coming in from outside the cell is going to cause this halt in the repolarization of the cell. The cell remains positive for a little while. That's the plateau. Eventually, however, the calcium channels, which open slowly, are going to close. This is the influx of ions causing the plateau. Then the calcium channels close. This whole while, the potassium channels have remained open. Once the calcium channels close, the potassium continues to be lost, making the cell more and more negative, and the cell finally reaches its resting state. So this is the contraction point. Contraction will happen after this point. So the contraction is happening right here. So that's the contraction. So compared to skeletal muscle, cardiac muscle contraction lasts longer. So it's not as quick a contraction as skeletal muscle has. And that is due to influx of calcium creating the plateau. Okay. So contrast now this curve that we see here with the plateau with the kind of curve we're going to see with the nodal or conducting cells. These are the SA node and the AV node and the AV bundle, etc. Remember that this is what creates what we call the autorhythmicity of the heart. The fact that these cells create one action potential after another, after another, after another. There is no resting state for these cells. So what we see is these continuous action potentials being created. At the very end, the action potential, the depolarization, is created by sodium channels that are opening and bringing in sodium ions, depolarizing the cell, making it more positive. However, most of the depolarization that you see here is going to be caused by voltage-gated calcium channels that will be opening, and it will be mainly responsible for the depolarization of these cells. So contractile cells are depolarized by sodium. The nodal cells are going to be depolarized by calcium. All right, so we have a calcium channel opening, which causes the depolarization. Once we get the peak of the depolarization, now voltage-gated potassium channels will open, and it will cause the repolarization phase. So here you have the voltage-gated potassium channels open and making the cell more negative until it hits back up to resting potential, except that the cells are not going to be at rest. Instead, as soon as they hit that resting potential, another action potential immediately starts. This is what we call the pacemaker potential, the continuous creation of an action potential. So that what you see are these curves that are constantly being formed. So these are just the continuous action potential that are being created by the conducting or nodal cells. Now, the fact that calcium is responsible for the contraction of these cells tells us that calcium ions are going to be the ones that are going to create or set the rhythm of the heart. All right, let's look at how the SA node generates these action potentials at a pace of 70 to 80 beats per minute. Now, the actual resting rate of a human heart is lower than 70 to 80 beats per minute. It's more in the range of 60 to 70 beats per minute. So there must be something is slowing down the SA node. That something is the parasympathetic nervous system. So the SA node has parasympathetic innervation via cranial nerve number 10, the vagus nerve, which is constantly slowing down the SA node from 70 to 80 beats per minute to an actual resting state of 60 to 70 beats per minute. If we were to cut or sever the vagus nerve, the heart would not stop beating. Instead, it would just beat a little faster, 70 to 80 beats per minute, because now there will be no parasympathetic tone is slowing down the SA node. It is worth noting that if something were to happen to the SA node and it stopped working, now the AV node will take over. So now the AV node takes over, And the AV node, however, has a slower rate of contraction. It contracts at 40 to 60 beats per minute. If the AV node stops working, now the AV bundle will try to take over. And however, its beats per minute will be about 30-40 beats per minute, which is not compatible with life. Typically nowadays, if the SA node stops working or doesn't work properly, we give the person an artificial pacemaker that will continue to set the pace of the heart. However, it is important to know that any of the nodal cells could try to take over. When they do, they produce what's called an ectopic focus, which is an abnormal condition in which you have AV nodes or other nodes trying to initiate action potentials. This happens when the SA node does not work properly or when there is some type of damage between the conducting pathways. This will change the heart and rhythm, and it will be definitely something that would have to be dealt with. Nowadays, with an ectopic focus, what they do is they just burn the little nodal cells that are causing the problem, and then the problem is solved. All right. The way that we could change the heart rhythm will be by, for example, stopping calcium ions from going into these cells. If we block calcium gates through calcium channel blockers, now we're going to slow down pacemaker cells. And with that, because remember, calcium is what's causing the action potentials in the pacemaker cells, that we will slow down heartbeat and slow down blood pressure or lower blood pressure. So calcium channel blockers are blood pressure drugs that are given to lower blood pressure. And the way these drugs work is by blocking calcium channels in nodal cells, which are going to slow down pacemaker cells and slow down the heartbeat. By the same token, the sympathetic nervous system has a profound effect in the heart. The hormones, epinephrine and norepinephrine, and are sympathetic hormones, and the neurotransmitter norepinephrine are both going to open calcium channels. In doing so, they're going to increase the rate of action potentials in nodal cells. It's going to increase the heart rate, and they're going to do more than that. They're going to increase the force of contraction or contractility of the heart. So sympathetic nervous system is going to have two effects in the heart. They're going to increase the heart rate and increase the force of contraction by opening calcium channels. This is what we call an ionotropic effect. All right, so that is important to remember, and we will go back to these here in just a second, and the next PowerPoints actually will do that. All right, let's see, almost finished with this set of PowerPoints. What the last things we need to look at is what we call the EKG, an electrocardiogram, which is a recording of heartbeat. And this is what it looks like. Needless to say, you have to know it, and you have to know the name of the curves and what they represent. So if we put electrodes throughout the heart and begin to make a recording of heartbeat, this is the types of curves that we're going to get. The first curve that appears is called the P curve, and it happens when the atria are depolarized. Following the P curve, there is this deep curve called the QRS curve, which represents ventricular depolarization. Because the ventricle has such thick muscle, it will create a much more pronounced curve. In the QRS curve, we have the T curve right here, which represents ventricular repolarization. So the atria depolarizes and then repolarizes. Then the ventricles depolarize and repolarized. Notice that ventricular repolarization is not seen in the EKG. the reason why it occurs at the same time as ventricular depolarization. So because the QRS curve is so large, it hides the atrial repolarization. So it is not seen. It is happening, but we don't see it. Okay, so atrial repolarization will be masked by the QRS curve. So PQ interval is the interval between the P curve and the Q curve, and that is showing you atrial depolarization, atrial repolarization. So both events at the atria. On the other hand, the QT interval is the interval between the QRS and the T waves, right here, and that is showing you ventricular contraction and relaxation, meaning ventricular depolarization, ventricular repolarization. The actual contractions happen right after the curve, the peak of the curves. So atrial contraction would happen right there, ventricular contraction right there. This is the delay between one contraction and the other contraction. This is showing you a normal EKG right there. Oh, sorry, that's heart block, because you can see this is one QRS curve, then there's nothing happening, meaning there's something preventing or delaying the movement of passageway from 1P curve, the atria, into the ventricle. So that delay is due to a block. This is a premature ventricular contraction, okay, right there, premature ventricular contraction right there. This right here is showing you a block. In this case, the block is in the bundle branch, which creates a prolonged QRS signal. So I'm going to go back. This is a complete a defibrillation of the atria. Atrial defibrillation. This will be ventricular defibrillation. So let me just go back here. Okay, actually, let me go back to there we go, this one right here. So if there is a block between the atria and the ventricle, so the signal will take longer to travel. If there is some type of blockage, let's say some cells that have died, or some blood vessels that are not delivering oxygen to the area, causing again cells to die, this interval right here will be increased because it will take longer for the action potential to travel from atria to ventricle. So that would be the delay that we're seeing. If there is the blockage in the atria, that would prevent the movement of the AP within the atria, then the delay would be between the last T curve and the next P curve. So that would be an atrial block. Okay. You should know the normal conditions, the normal EKG, and the parts of the normal EKG. Tachycardia is what we call when the heart beats in excess of 100 beats per minute. Remember that normal beat was between 60 and 70 beats per minute, so 100 is pretty high. On the other hand, bradycardia occurs when heartbeat is less than 60 beats per minute. An elongated PR interval is showing you a delayed conduction within the atria. So that tells you there is some type of blockage of blood vessels or an enlarged atria, which makes it the AP action potential. It takes it longer to travel through the atria. An elongated QT interval indicates an abnormal conduction through the ventricle due to an enlarged heart or through a heart attack that has destroyed some of the cells. We already talked about calcium channel blockers and the epinephrine or epinephrine actions. Okay, so I believe that is it for this first PowerPoint. Let me just go ahead and, yeah, that's it for that first PowerPoint. I'm going to go ahead and stop the recording now, and I will go over the second